So I'm going to talk about the journey we took to find the causative agent of sea star wasting disease. I'm Alyssa Gehman. I'm the one who's talking to you today, but I wanted to highlight that Melanie Prentice is somebody who's collaborated with me on a lot of the work I'm going to talk to you about, and also collaborated and made some of the slides that are going to be in this presentation. So yeah, so as maybe most of you know, in around 2013, we started to observe massive die-offs in sunflower stars. And so these are these incredible photos that were taken. This is early in October in 2013. You can see that there's just piles of sunflower stars here. To the trained eye, there's actually some sunflower stars that look sick. Like this one here has some twisting that looks to me like it might be at the early stage of wasting in this picture. But we wouldn't have known what that meant at the time. But what we did find out was that later in October, just a few weeks later, there was massive die-off. So we went from having just the whole ground covered in these sunflower stars to none of them, which is an incredible and really upsetting change. And this happened not just, in that one location, but after we sort of saw those observations. This is a time lapse. On the left, this is places that people have looked and there was no disease. We're just sort of slowly going through time in 2013. On the right, these points are all places where we are seeing disease. And so you can see that like by November of 2013, we've observed disease really pretty far up and down the coast, like all the way down to Mexico. Now we're starting to get a little bit further north as we're in 2014. But yeah, I mean, we don't have to memorize the timeline of this, but I think the main thing to note is that this spread quite rapidly over a very, very large area. And this resulted in massive loss. So as Sheila said, we around, I guess this was 2020, 2021, we put together a data set of what we knew about sunflower stars on this coast from Mexico to Alaska. So that was a big collaborative group of researchers. And then we broke the coast down into little sections and asked, okay, how many sunflower stars did we lose? And each section in color is a subsection of the coast. And the number written there is the percentage of the populations that we lost during this disease outbreak. And so you can see, I mean, as you go further south, that's upwards of 100% in some locations. And then in the BC area, we're relatively low with our 87.9% loss. And so across the whole coast, this is a 90% loss of these species. The rough estimate was over close to 6 billion sunflower stars. So just an absolutely devastating and incredible loss. And at this point, even in 2022, we really didn't know why. Like Sheila said, there was other researchers who did work and thought they'd found a virus, but then later disproved themselves. And found that it was not, in fact, the causative agent, didn't cause disease in sea stars. And so around 2020, as we were putting this together, my research group was asked, what's the next question that we need to answer to try and save sunflower stars because they're critically endangered? And my answer at the time was just, we have to know what's killing them. It's incredibly hard to know how to save a species if you don't know why they're dying. And so there's a bunch of reasons that we might want to know why they're dying. I mean, just because it's a great mystery, but also, and it could be because they're incredible animals. These are because they are incredible animals. If you look at this photo really closely, you might see tiny little white dots sticking out of the top of the skin. And if you look really closely, I'm wondering if I can circle it, you might actually notice that some of them are fork shaped. These are actually little pinchers. So their entire dermis is covered in tiny little pinchers, and they just pinch you if you Like if you have ones these big, if you have it next to your arm, it grabs your arm hair. So they're, I mean, that's not a reason to save them maybe, but it's they're an incredible animal just in their own right. But they are also massive predators. So if you look sort of in the distance around these sea stars, you can see that there's urchins, all kind of not right where they are. They're all just like a little bit away. And that's not it's not an accident. This is another photo of sort of these big, beautiful animals. And I actually want to take a moment because I know when I look at this picture, I can kind of get a sense of size, but that's mostly because I actually know what size they are. But I think I'm going to stop share so that you can see me. And I want to just show you like this is the size of a sunflower star. So like, and this is a normal sunflower star. This isn't a big one. This is just a normal sized sunflower star. They're really, really big animals. And they're major predators. So I'm going to jump back in. Here we go. And if you look kind of in the corner over here, what you see are piles of urchin tests that were likely eaten by all of these big predators. And so what we found when there was this massive die-off where we lost 90% of this population, and they really do, they can be quite abundant when they are there, is that they are actually quite important for maintaining kelp forests. So you probably already know the story of how otters are really important for maintaining kelp forests because they eat urchins. It turns out that sunflower stars also eat urchins, and they just eat a different size of urchin and quite a lot of them. And when you look at sort of the comparison of otters and and sunflower stars on urchin populations, sort of the healthiest kelp forests were in areas where you have both predators consuming the urchin population. And so what we found was after this big die-off, big die-off of sunflower stars, there was a mass proliferation of urchins and a big die-off of kelp forests. And this happened in many places along the coast, the most drastic of which was in that area in Northern California, where we have 100% loss estimates for sunflower stars. And so even beyond just how cute and wonderful I think the sunflower stars are and how sort of incredible they are on their own, they're really important for maintaining the ecosystems that are around them. Okay, so, that's why I think we should try and save them. And when asked what we needed to do to save them, my answer and my team's answer was we need to figure out why they're dying. And so this group of researchers here, I'm on the left, and then I'm with Grace Crandall, who's a graduate student at the University of Washington, Melanie Prentice, who's a researcher with me at Hakai Institute, and then Drew Harvell, who was at Cornell and is also at Friday Harbor Labs at the University of Washington. We, plus a very large group of other people, worked together to start running experiments to address this question. And so the first thing you do when you don't know what is killing a bunch of animals is attempt to control the passage or transmission of the disease. So we know that a bunch of things are dying. We know they are dying in a somewhat similar way. So I mentioned the twisting. There's twisting, there's arm loss, there's lesions, there's things that we see in the field. And that suggests pathogen. And so one of the things we can start by asking is it something that's transmissible? Is it something that we can control? And so that's what we do for these disease challenge studies. So we start, and I think maybe the thing to remember at the beginning here is that we have no idea why they're dying. And so the goal is to design something that is, how do I put this, like agnostic to the answer. So we want to be as open to what could possibly be killing them as we can be. And so we start by collecting animals from the field. And at this point, post the outbreak, we do have some sunflower stars to collect. And And when we do, some of them will show signs of disease. So we would collect from the field. Actually, we mostly collect animals that look healthy, but once we got them into the lab, a couple of them would show signs of diseases. So this one here is showing twisting, and sort of these twisty arm bits. And so what we would do is collect tissues from that animal, and then we would have two different treatments. So we'd take those tissues, and the assumption is, because this is a sick animal, that if we have tissues from that sick animal, then the pathogen or the putative pathogen would be inside that tissue. And so we divide that tissue. And in this case, for the most part, I'm going to talk to you about something called coelomic fluid, which is the body fluid or kind of the blood of a sea star. And what we would do is we would separate that fluid into two different treatments. One, we'd keep cool, and hopefully the pathogen would stay alive and active, and we call that the exposed group. And the other, we would boil for 10 minutes, just like you would if you were wanting to drink water from a stream and would like to not have the bacteria or pathogens get into your body and get you sick. We'd boil it, and so then we would be deactivating. If there's a pathogen in there, we're deactivating that pathogen in this control group. And so we take those two different treatments and we put them into animals that we know don't currently have the disease. And so that's actually something I should take a moment to explain. One of the challenges for running these experiments is that the disease is in the field. Remember, I said that the way we started this experiment was we went into the field, collected animals, and some of them got sick. And so in order to be sure that they weren't already sick, every animal that we used in an experiment, we would bring them into the lab, we would isolate them and quarantine them for 2 weeks and watch them and make sure that they had no signs of disease during that period of time. And it was sort of, as an aside, this was all happening in like 2021. So it was sort of an interesting parallel where we'd, somebody be isolating for COVID for 2 weeks and we'd isolate our sea stars for 2 weeks. It was a very similar situation. And at that point, we would know that they don't have the disease already. And then we would use them and expose them to either control, which should have nothing, it should be deactivated, or this exposed group where we think that there's a pathogen inside. And so then we would watch them and see what happened. And what happened to the exposed group is that they all showed signs of disease. And they did so about 3 to 12 days after we'd exposed them. Unfortunately, it's a very upsetting disease. So they sort of twist, but then their arms drop off, and it's quite a thing that would happen to all of the ones that are exposed at a slight delay to when they twist. And then importantly, none of our controls ever showed any signs of disease. So if we deactivate whatever is in this fluid, we can inject it into a star and they're fine. And so that's a really important differentiation. I'm going to be showing you results from this particular experiment with the numbers that are here, but we actually ran experiments like this over 20 times. So we repeated all of the experiments that we did, and we confirmed that we were getting similar results as we did our work. And so not only did they not show any signs of disease in the control groups, we also had no mortality at all. And we had 100% mortality in our exposed group. And I just, to say, this is not the kind of data that you normally get when you're running experiments. Normally there's some variability here, but it was very clear. Nothing in the controls died and everything in the exposed died. And that kind of matches to what we saw in the field, remember, because we would go and we would see some animals, we'd see a bunch of animals start to get sick, and then you'd come back and all of them have died, right? So this sort of incredible pathogenicity, this incredible mortality that we were seeing in the lab, we're also seeing in the field. Okay, so those experiments, I was talking about coelomic fluid. I'm going to show you results from a whole range of experiments using different ways of transmitting disease. So same kind of idea, we just use different things to do the transmission. And before I start showing you the data, I want to show you the 3 different stages that I think I've talked about. We have down here apparently healthy, which is just basically with your eyes, does it look okay? And these 2 sea stars that we have pictures of, those look like happy sunflower stars. Then we have diseased with arm twisting. And so you can see this one here has a bunch of arms twisted, and this one has a bunch of arms twisted here. And then the next stage is disease arm autotomy. Autotomy is the term for basically losing your arm on yourself. So nothing pulls off the arm, the sea star just releases the arm. And they have a biological process for releasing their arms or dropping their arms. And so you can see there's an arm here and an arm here. And yeah, I will say that it's somewhat shocking and upsetting to see this happen, because the arm will actually walk away from the body, as will the sea star. It's gruesome. Anyways, so what we're going to look at on this graph is first, on this side, we're looking at wild source transmission. So these are starting from animals from the wild, like I described earlier. This axis here is time. So 0 is when we started, that at the beginning when we do those challenges. And then every day after is, a day or 15 days from when we expose them. And these are the 3 different stages on the bottom. And so the first thing I want to show you is we did transmission trials where the sea stars had actually no contact at all with each other, but we had a 6 star, a line that let water go between the 2 containers, and then a recipient star is the term we use, that would get that water. And we would see, okay, can the disease transmit by water? And the answer is yes. And you can see this trajectory. It takes about 10 days before they start twisting, and about 2 weeks before they start dropping arms and dying. The next one I'm going to show you is raw homogenate, which actually just means a bunch of different tissue types all ground up together. So for this one, it's, we're not honing in on anything specific. We're just getting a bunch of different types of tissue, just in case, if you're thinking about when you don't know where the disease is in these animals, which we didn't, if this was strep throat, we wouldn't want to just get their leg, because their leg probably wouldn't have any strep in it. So this is raw homogenate, and the different colors here are just different years. So we ran these experiments. Like I said, we repeated them over multiple years. And so you can see we had twisting, followed by dropping, followed by mortality. Again, sort of this same trajectory of disease, but this time a little bit faster, which suggests that there's potentially more pathogen in this tissue. And then we decided that we would check and see if the pathogen was in that coelomic fluid or body fluid. And you kind of already know the answer to this question, but it is. And you can see that, again, that the trajectory of these disease signs is the same. But now when we use coelomic fluid, it's a little bit less than 5 days before we start seeing twisting, and mortality is less than 10. So it suggests that the pathogen is actually in higher concentrations inside that coelomic fluid. We're putting more in when we use that. Okay. So, and this is a picture of Melanie Prentice, who I mentioned earlier. This section is very much the section that she led. The next question is, clearly there is something in that coelomic fluid that's causing disease. What is it? And so we did 2 different genetic techniques. The first one is metatranscriptomics. And the cool thing about metatranscriptomics is that it looks for anything. Like, it's basically a method that says, what is inside give me a list of all of the things that are in the coelomic fluid. And so we got a list of all the things. And it turns out to be quite a lot of things. So like the coelomic fluid is full of stuff, and so are we, right? We have lots of bacteria in and on our bodies. So do sea stars. And so you can see there's lots of things that were in that list. Some of them are bacteria, but there's also eukaryotes, archaea. There are some viruses in there. There's all sorts of stuff. But the next question is there something unique in that coelomic fluid compared to between the controls and the exposed? Because we see really different responses. What is it that's unique about those? And so that's this figure here. And the way to read this figure is any point that's nearby each other. So for example, these here is more similar. Anything that's far apart on opposite sides is more different. And all the points in yellow are individuals. This is individual samples of coelomic fluid that don't have any disease. So they came from something without disease. Everything in blue was dying or of disease. And then this little star is actually that inoculum that we used to put into them. So like that's what we injected in. And so what you can see is that there's a big difference, right? The blue is far away from the orange. And so the next question is, what's driving this difference? Because this isn't giving us anything specific. And this is when we were actually quite surprised. The answer is one thing. So there's 3 arrows here, but Vibrionaceae, Vibrio, and Vibrio pectenicida are essentially 3 different ways of saying Vibrio. And in particular, this one species, Vibrio pectenicida, which I'm going to refer to as VPEC for the rest of this talk because it's easier to say. And so this, again, kind of to repeat myself, this is not what we expected, or nor do you normally have one thing that's different. Like microbiomes are variable. I mean, we expected there to be some variability just generally, but overwhelmingly and strongly, the thing that was different between our controls and our exposed was this one pathogen. And the nice thing about VPEC, which is a bacteria, is that means we can sequence it and do a different kind of sequencing that's called 16S, which is cheaper and easier and faster than the first thing that we did, metatranscriptomics, which gives us everything. And so we did that next. We said, okay, now we can use 16S. Excellent. Let's look at our same samples using 16S. And I mean, maybe this isn't shocking, but again, Vibrio pectenicida VPEC was the main thing that was different. And this is another way of looking at that. So what we have here is the relative abundance of VPEC in the list of, that list of everything, in this case, the list of all the bacteria. And we have this is all the stars before we did anything to them. So before we started an experiment, we would sample everybody. And so what this says is that before we started, there's no VPEC in there. And then after, when we have our diseased ones, this dark blue dot is the mean. And then all the light blue are the individual points. So this is an individual star and all of their individual amounts of VPEC. And what you can see quite clearly is that the control group is down here at the 0 area. And while there is variability in the percent reads, all of the exposed group has quite a lot of VPEC in there. And then again, just for confirmation, what we put into them to cause the disease also has a lot of VPEC. And so the amazing thing about bacteria is that you can culture them. So you can bring them in and grow them in the lab. And you can grow them and then you can isolate just one type of bacteria. Because previously, when we were putting in that coelomic fluid, yes, we were putting in VPEC, but we were also putting anything else that was in the coelomic fluid in, right? And so what we would like to do is ask, if we put just VPEC in, what happens? And so to do that, the first thing we needed was a sample that had VPEC in it. So this might be counterintuitive, but when we got this result, it was not a time when we were running experiments. It's not a time of year when there's generally disease in the field because there's seasonality in the timelines. And so we actually didn't have a straight up forward way to get samples of VPEC from an animal. But luckily for us and unluckily for our collaborator, they had an unseasonable outbreak of sea star wasting disease in their labs. Remember, we've shown that the disease can actually transmit through lines. And so they had some disease come through the lines and get some of their sea stars sick. And they called us and let us know. So we drove down and collected coelomic fluid from 2 animals. You can see that they're twisting. You can also see this was at night. They told us this was happening, and we basically got in the car and drove immediately. It was sort of chaotic. Anyways, we get down there, we get the samples, and we are able to culture those samples. So this plate is what a Vibrio pectenicida VPEC looks like if it's growing on a plate. This was done by Amy Chan at the University of British Columbia. And this, you don't need to be able to read this big long sequencing line. If we zoom in, basically we did the genetics. We cultured this thing that we got from these sea stars that were wasting at Friday Harbor. And lo and behold, what we cultured was VPEC. So, and remember, this is like an entirely different time, from a different place, from a different outbreak. So, and we did actually run these experiments over multiple years, and every year we ran the experiments when an animal's sick, VPEC was, we got that animal, the animal had VPEC. So it's starting to kind of add up in evidence that it's what's happening in the field as well. Okay, so now we can go back to that control challenge experiment that we first introduced, but we can use cultures of just VPEC, nothing else. We know what it is. We know how much is in there. So that's one of the other things that was really exciting, is we can now actually set doses, because we couldn't do that when we didn't know what we were working with. So we went back, and instead of taking tissue, we just started with cultures, and similar, well, basically the exact same methods. We heat killed half of them, didn't heat kill the other half, exposed animals, and watched what happened. And this is sort of a table of the response. The main thing to get from this is, and the doses are how much VPEC we put in, because I told you we can control them. So 10 to the 7 is quite a lot. And then we sort of scaled down from there. But essentially, we had almost 100% mortality. We had 100% mortality in the 2 highest doses. In our lowest dose, we had almost 100% mortality. And so we're really seeing essentially the same response. So now this is confirming that VPEC on its own is a causative agent of these sea star wasting disease signs. And then just to show you that kind of this pattern of twisting, then dropping, and then mortality that we saw when we were using wild sourced of disease is also shown in our cultures. So this is that lowest dose. And you can see the lowest dose is actually takes a little bit longer to cause disease than our coelomic fluid doses did. And then as you go up in dose, you can see sort of the disease progresses faster and faster. So yeah, so that is where we got to find the causative agent. This is the work that we did to do that. And the thing that I should say is that, VPEC existed. We didn't name Vibrio pectenicida. It was a name that was already assigned to a bacterium. And so one thing that's been really interesting and is a source of future research is that the original VPEC was isolated from scallop larvae in France. And so it causes disease in scallop larvae and in oyster larvae. And what we know is that disease or the mortality in scallop larvae is driven by toxin production. So one of the things Vibrios are known to do is produce toxins that kill your host. And so we don't know yet what the mechanism of mortality is for VPEC in a sea star, but the fact that VPEC in scallops is caused by a toxin could suggest that that's a pathway that is possible for VPEC in sea stars. And when we look at the genome of VPEC in a sea star, there are some things in there that look like toxins. So that's future work, but we will see. So everything I just told you about, we were collecting from the field, we were doing sampling from the field, but there was a lot of work that is in the lab. And so the next question we had was, okay, during an actual disease outbreak in the field, do we see VPEC? And so luckily, myself and my dive team up at the Hakai Institute have been out doing sea star surveys since 2013, but we've been doing targeted ones since around 2018, I believe. And we started collecting when we started noticing that coelomic fluid was an important way of finding VPEC. We started collecting coelomic fluid from animals in the field. And so this is a data set that comes from nearby Calvert Island. So just to orient you, this is Vancouver Island here. We're on the central coast of BC. And then we're actually in two fjords that are nearby here. And we went out in May and did surveys. And in those surveys, we found sunflower stars, but we didn't find any wasting. So all those points are black. There's no wasting disease detected. But we did collect samples from the animals that were not wasting. We went back in October of that same year to the same places. And now you can see two of the sites. We observed wasting disease in the sunflower stars, and several of the sites we didn't. And so we collected samples from here. And for how I talk about these, we're going to have sites where there was animals that were not affected, like we didn't see any wasting disease there. And then at the sites that had wasting disease, we have animals that looked okay and animals that looked like they're wasting. And one of the things I want to set you up to remember is that in all of the data sets that I showed you in the timelines to signs of disease, the shortest between when they were exposed to disease and when they started showing signs of disease was like 4 or 5 days, but the longest was 10 or longer, right? And so, and honestly, the experiments we're running, we're probably giving them pretty high doses of the pathogen in the lab. And so we might expect that animals that look okay, that are nearby animals that are sick, could just be in that lag phase. So they're in a time period where they don't look sick, but they might actually still be harboring the pathogen. And so here we are. We've got, again, sort of that same kind of graph where we have relative abundance of VPEC on this y-axis. And then this is May when we went out. And in May, when we saw no signs of disease anywhere, there was no VPEC. So those are zeros, no VPEC at all. In October, when we came back, we found VPEC in some individuals, almost 100% of the reeds, especially this one up here, were full of VPEC in the diseased animals, and this is at sites that have diseased. We also see large amounts of VPEC in animals that look okay, but that are at sites that have disease. So again, probably the individuals that are in this kind of lag phase. And then at the sites that didn't have any disease, so at the unaffected sites, we saw very little or no VPEC. And so this is very supportive of what we're seeing in the lab is also what we're seeing in the field. And we'll get into this a little bit later, I think, but one of the things to note is, if we had known to look in this coelomic fluid in the blood of the sea star for these pathogens, this kind of data is clear enough. If we could have just sampled the sea stars in the field, But when you don't know where the disease is, if you look in the wrong tissues, it doesn't show up as clearly as this. Okay, so that was all from Central Coast of BC, and it was from in the 2022, I think. We wanted to look further back in time, and we actually are continuing to look further back in time. But luckily, one of our collaborators has this data set that's from 2016. It's from Southeast Alaska. We're zoomed in quite far here, but the main thing to note is that this green area is like quite similar to what sort of the time point that I saw today. But this was, to the best of our knowledge, the front end of the disease spread. So this was the time point where no one had seen any disease in sunflower stars in 2016 at this green site. So we consider that uninfected. The disease had not got there yet. But 70 kilometers south, we have wasting disease. And so then down here, we have, again, sort of affected sites with grossly normal. Grossly normal just means sort of the same as apparently healthy. It's just, it looks okay on the outside. And then ones that look like they're wasting. And so this is the work that they did. The main thing to note is from the affected site, both grossly normal and wasting animals had a lot of Vibrio naceae. So that's in this orange. And their data set didn't make it easy for them to get below that, but it was, suggestive. And so since we know that VPEC is something we're interested in, we went in and queried this same data set and said, well, is there any VPEC there? And the answer was there's lots of different Vibrio species. There was VPEC. And if you look across these samples, 6% at the unaffected site had VPEC, but 60% at the affected site and 67% of the wasting stars at the affected site had VPEC. And so again, this is showing us that VPEC was present in these wasting areas in sort of consistent patterns at the earliest stage of the spread in this region. So just to sort of summarize on a map what we know so far is, we did our experiments in Washington, and so we know that the wild animals have VPEC in Washington because that's where we got the pathogen from. We know from the sort of extant disease outbreaks in the Central Coast that VPEC is present there. And we know that it was present in the earliest of the outbreak up in Alaska. We are still working with colleagues to get access to samples to look in California, Washington, and Oregon. Well, not Washington we have, but Oregon and California and Mexico. It's a lot harder to get anything recent because so many of them died that there's not really any animals to sample. Okay. All right. I might do this section quickly, given the timing. But the next sort of research going forward for the future is, all right, sunflower stars have VPEC and it's associated with sea star wasting disease. You likely know that there are lots of other sea stars in this region and that other sea stars were affected by sea star wasting disease. So what do we know about VPEC in the other sea stars? And this is, like I said, ongoing work. So first, I'm going to talk to you about some experimental work that was led by Grace Crandall from the University of Washington. And this is where we took sunflower stars, ochre stars in the middle here, and then leather stars. And these are three different stars that kind of are on a scale of resistance to sea star wasting disease based off of field observations. So there's a lot of the leather stars seem to have actually, increased in abundance in many places during the disease outbreak. So they seem to be quite resistant. The ochre stars definitely got wasting disease and had big losses, but have seemed to rebound a little bit faster. And then sunflower stars, which are sort of the biggest losses, slowest rebound on populations. And so what we did is we put all three of these species into a container together. And we put a fourth animal in, a fourth sunflower star that had been exposed to disease on one side, or is that control? So similar to the control challenge trials that I showed you earlier. And so we called this sort of the focal disease agent. So this is going to be a disease star. It's in there and it's transmitting to its fellows. And so the question is, can a sunflower star transmit to a dermasterius or to a pyesaster? And then the controls, this one should stay healthy and everybody should be fine. We actually ran this experiment for 59 days. So it's a really long experiment. And I don't think I'm going to show you any data, but what I am going to tell you is that the sunflower star that was in here, both sunflower stars, they all died, as did most of the ochre stars and none of the leather stars. We were there for 59 days. Every star around them died, and the leather stars just kept on going, which was incredible and is something that we're going to continue working with and understanding. And one of the really cool things is that Grace is doing her PhD in transcriptomics, which is the study of, well, in her particular case, the study of the immune response of these animals. And so we're hoping to be able to look at what each of these animals are doing and see, like, are leather stars resistant because they have like a cool way of fighting off the disease, or are they just not susceptible at all? Like, what is happening and why are these guys different? But to the point of, is there any VPEC in there? The answer is yes. In ochre stars, in the wasting ones, they all had VPEC and large amounts of it. Interestingly, in the controls, they also had VPEC in them. And this makes some sense, because one of the things I didn't tell you is that we actually had one ochre star that got sick, it looked terrible. It was all twisted up and pale, and then it got better. And that was incredible. We hadn't seen that in sunflower stars basically at all. But that is really suggestive that these ochre stars do have some resistance. They can actually not die when infected by this disease. And so it would make sense that you might find some animals that look fine that have a little bit of VPEC inside them under those circumstances. But certainly there's large amounts in the ones that died. And so then that's one other species or two other species, but there's tons more. This is a very sad solaster. And so the question is, what about the rest of them? And my answer is that we're working on it. We've been sampling animals in the field for the last bunch of years and are starting to look at these samples. And I'm just going to give you a little preview. This was the thing I did promise to tell you about, and that was that what type of tissue we use, we found really matters. And so this is the fluid that I was talking about, the body fluid. Tube feet are just those little feet at the bottom of a sea star. And then dermal swabs it's kind of like the COVID swab in your nose, but it's just same swab, just on the top of a sea star. And so if we're looking these are data from the lab where we know that they're dying from wasting disease because we exposed them. If we look for VPEC there, if we look in the coelomic fluid, it's 100%, it's always there. Tube feet, 75% of the time. So in animals that are definitely dying from wasting disease, we don't always detect VPEC in their tube feet. And similarly, we higher than the tube feet, but dermal swabs are lower than the coelomic fluid. So if you really want to be sure to be able to detect it, if it's there, you need to look in the coelomic fluid. And even more importantly, the average abundance is even bigger difference. So in the coelomic fluid, the average abundance is very high. It's predominantly VPEC when they are dying from disease. But in the tube feet, even if it's there, it's less than a percentage. So it would be really hard to detect VPEC from a tube foot sample, and it'd be really quite probable that you would miss it. And then again, dermal swabs are kind of in this middle ground where they're not quite as sensitive as coelomic fluid, but they're not as insensitive as the tube feet are. And so for the field work that I'm going to talk to you about, it's in this dermal swab category. So it's not as, and we now have coelomic fluid going forward, but what I have to show you today is dermal swabs. And so this is what dermal swabbing looks like. You can see the swab on the sea star. This is some of my research team. So this is Jillian Sadlier-Brown, Derek Van Manen, Zach Monteith. In this picture, there's somebody else there, but I can't tell just from their hat who they are. And we actually have this like mobile lab on our boat that we use to take these swabs. And so first, just to show you, if we're looking for VPEC on the surface of Pycnopodia, this is the relative abundance of reeds down here. And the colors are how sick the animal is. So first things first, if a pycno is wasting, we do detect VPEC on the surface, as we would expect to. Here's some ochre stars, which I've already shown you. There is VPEC associated with wasting in the lab. You can see that they're sort of basically, as you go up in color, you're getting more and more diseased. For ochre stars, you need to be kind of in this very diseased state for the reed abundance on the surface to really start to show up. And then interestingly down here, this is the mottled star of Asterias troscelli. And I think it's really interesting that as you go, sort of this is going more and more diseased up in these colors, you see more and more VPEC in those reeds. So again, really suggesting that VPEC is causing disease there too. Dermasterias, as to be expected, we don't really see signs of disease. We do see a little bit of VPEC. All these little points are just kind of, they're outliers, but there is some VPEC in there. We do see some in Dermasterias, which I think is also true from the ones in the lab that didn't get sick. This is from a relatively low number of samples, but we do see VPEC in Solarster stimsoni that are sick. And then I think Stylaster, we have more data than that's here. So I wouldn't take this to mean that it's not there, just that we haven't run enough samples yet. But this is just to say we are seeing it in other species. And then kind of just as a fun aside, if we look in the seawater, you can see sort of there are detections of VPEC in seawater. And this is just, basically an Nalgene of water that we collected while we were diving. And so that suggests that we could potentially use Nalgenes of water to get a sense for how much VPEC there is in an area, which might be really useful for when you're trying to think about how to outplant animals, for example. Okay, so I like to just note that all of the diving data that I showed you was done by these divers, including myself, but it's thousands of hours underwater. So it's kind of an incredible resource that we have at the Hakai Institute to have these professional divers that are able to go out and do this kind of work. And I think I'm going to just spend a little bit of a moment telling you a bit about the Hakai Institute, and then we'll switch over to questions for the talk. So I'm a researcher, principal investigator at the Hakai Institute. Hakai has 2 field stations that are highlighted here. The Calvert Island station, which is where a lot of the data that I talked to you about, we were staying here on the Calvert Island station when we did that work. And then the Quadra Island station, which is shown here. We have mesocosms and genomics labs and a bunch of resources available on Quadra Island. And Hakai, I've been talking quite specifically about one area, but Hakai does science on the coastal margin, and so we work across a bunch of different habitats and work together in teams to try and address questions about how our ecosystems are doing. Yeah, and this is just some examples of other types of work that we do. Some of you may have heard of our Light Trap program, which is a program that's looking at recruitment of Dungeness larvae. If that's something you're interested in, I highly recommend Googling the Sentinels of Change, or I think that's the easiest way to get to it. And these are all projects that are part of that. So we have sort of experimental tiles that are out there. These are some subtitle arms that we put out to look at recruitment devices. This is some researchers doing surveys and transects. Actually, this is just in West Van. Yeah, and so this is a selection, not everybody, but this is a selection of folks that work at the Hakai Institute who make all my research fun to do, and a bunch of folks that we collaborate with on this kind of work. And yeah, I want to thank you guys for listening, and I think I'm ready to take questions. I think I'll close out my share screen unless we decide we need it later. Oh wait, did I already do that? Oh, there we go.
Awesome. No, you're still sharing, but there it's gone. Amazing. Just so much work involved in all of that, but I can see how it's very logical. I mean, in the way you're approaching, which of course is pretty important. Um, I'm curious about the other examples that you used on the the other 1, 2, 3, 4, 5 sea stars that were showing signs. Joan, do you want to go to chat first or?
I absolutely will. We have one question so far in the chat. So, everybody, you can put a question in the chat, but we'll also unmute you if you wanted to ask questions. So, first of all, from Rosemary, Rosemary Cornell, we have two Rosemarys on tonight. Alyssa, great work. I assume that Vibrio has been present prior to the emergence of wasting disease. What factors in the ocean made the sea stars suddenly susceptible to the VPEC starting in 2012 to 2013?
That is a fabulous question. At the beginning of this talk, Sheila asked me if we were done with this and moving on, and the answer was no, because one of the things one couldn't ask until you knew what was causing sea star wasting disease was why now, why then. And so that is one of the questions that's very much in my mind. There's certainly a variety of pieces of evidence. We know that, shortly after 2013, there was anomalously warm temperatures, and there's some pretty good evidence from both the lab and the field that sea stars, when they are with sea stars and the disease mixed together, is very bad for the sea star in warm temperatures. So the disease and warm temperatures. I'm going to maybe restate that. Basically, there's an interaction between temperature and the disease. So like we have seen in the lab that at warmer temperatures they die faster. At cooler temperatures they die slower. We can now test whether a sick animal in a warmer environment produces a lot more VPEC and makes it more contagious. Or I guess there's like a couple of things that could be why we see more disease in warmer waters. And so that's work that we're starting to do now, is asking, why do we see more disease in warmer waters in this particular case? And as we answer that, it could be that something to do with temperatures, why then? It also could be Vibrios are known to be pretty changeable. So it's possible that VPEC was around, but wasn't pathogenic to sea stars. And something about it changed that made it a disease for sea stars in that time period. And so to answer that question, we need to try and get different strains of VPEC from different animals over different times and see how they've changed over time. It's also possible it could have been introduced. There's like, we really, I guess the short answer is we don't know. The long answer is there's lots of ideas, and we're going to start looking.
All right. I see, Peter, you have your hand up. You should be able to unmute yourself and ask your question. You can't unmute yourself, huh?
Oh, it went away for a second.
There we go.
Oh,
Yay.
oh, try it
Oh.
again. Unmute yourself again.
Oh,
There we go. You hear me?
yeah.
Yeah.
Oh, good. Since the bacterium, of course, was around before, is it possible that another pathogen could potentiate it?
Right. So like an interaction between two
Yeah,
pathogens could
because
be.
we heard other hypotheses before about funguses and viruses being involved.
Yeah. So, I mean, the virus hypothesis has been disproven by the people who originally put it forward. So it's probably not a virus. So I guess maybe interestingly, Vibrios can interact with other species. So it's possible that some other species was interacting with Vibrio and that changed its pathogenicity at that time point. Yeah. And you know, we need to keep looking at the evidence and see if we can find any additional evidence for that. But right now the evidence suggests that it's sort of this one pathogen, at least for now.
Can I just ask a confirmation that the Vibrio is actually a bacteria? Is that not correct?
Yes, it's a bacteria.
Yeah, okay.
Yes.
So Carolyn is asking, what is the pathogenic mechanism of this Vibrio? There are some other very destructive species like Vibrio vulnificus and cholera.
Yeah. That's yes. Vibrios are known for their pathogenicity. So great. Yes, exactly. Good point. And we're kind of at this point, we don't know what the pathogenicity is. But it turns out that for many Vibrios, there's multiple ways they kill their host. So they tend to have an array of mechanisms. That's one of the next questions is how is it killing the host? Our current evidence suggests it could be a toxin, but we don't know that yet. So we need to do more work.
Mm.
And when I say toxin, it's the Vibrio would be producing a toxin while it grows, just to be clear.
So that would be similar to, like, it would be like a poisoning, like E. coli produces a toxin that poisons you.
Yeah, as best I understand it. Yeah.
Okay. So Sharon was asking, is the fact that sunflower populations were so high, maybe overcrowded, that made them the most susceptible species, or was it something genetic?
Yeah. So they're certainly high and they're certainly overcrowded. I mean, the photos I showed you, there's lots of them. And they're big and they move a lot. So all of that is true, but that isn't necessarily why. Well, I guess it wouldn't explain the susceptibility in the lab. So like the fact that they all die and some of the ochre stars can survive. And so I think it's probably something maybe genetic that is different between the different species that's making them different amounts of susceptible. I do think that, you know, the fact that they all aggregate together is certainly not helping them. I will also say, as somebody who dives, and actually we see this in the lab too, if there is a wasting star, other sea stars will eat it, which is a terrible idea. It's a great way to get a disease, to eat other diseased things. So they're certainly not helping their case in the field. So like not only are they near each other, but as they die, they will eat each other. And I think about the fact that they have up to 24 arms, and the arms do basically all pop off and walk away. So you'll find them kind of all over the place, which is another potentially great way to just spread disease. So it could be all of those things kind of make them more contagious, if that makes sense. But that doesn't necessarily, it does also seem that there's something about them as a species that is just more susceptible as well. So it's probably a little bit of both, maybe.
All right. So Rosemary has a follow-up question. Wasn't there a discovery of some clans of sea stars recently on the BC coast that are unaffected by the wasting disease?
I think that's probably my work as well. And it's not quite that, but we did find, so the pictures I showed you are actually from 2020 in the fjords of BC. And so what we found was, and as you saw, we do find VPEC in the fjords of BC, but what we find in the fjords is that even though the disease is there, we don't see the level of mortality that we see everywhere else, basically. So that, that picture I showed at the beginning where you have like tons of them, a little bit of disease, and then all of them die, that's happened repeatedly in many locations. In the fjords of BC, we see some disease, but we don't see this massive population loss. And one of the things that stands out for these populations is they live in a specific area of the fjord that is cooler than everywhere else. And so I think that that's actually reducing how much the pathogen is able to grow. And so it's not that it can't grow at all, but it doesn't grow at such a rate that it kills everything. And that's that we're following up on that. But yeah, we do see. So it's not that they're not dying in there, it's just they're dying at much lower rates. And the fjords are refuges from this disease, just not in the no disease, there's not as much mortality.
Could I follow on to that? Because that when you showed the Alaska, the original location, I mean, the blob was off the coast. I'd have to look it up again to see how far northward it extended, especially in terms of the warmer waters. But it seemed interesting that I would have thought that cooler water was a help, and yet it did affect populations up there. So was it the heat blob, or was it just a particular location that could have made that initial outbreak more prominent? I mean, there may not be an answer to that, but I can see where the temperature could slow down or speed up the population effect, or the effect of the population.
Yeah. So one of the things that's kind of a nuance is that while temperature makes it worse, it isn't necessary for the disease, if that makes sense. So like, it doesn't have to be hot for there to be a disease outbreak. It's just if is hot and there's a disease outbreak, it might be a worse disease outbreak. And so I think, when we look at the like percent loss, we do see for the most part reductions in loss as we go north. And so that could be because temperatures are cooler. So we're still seeing massive, like there's still 90% loss, but it's not California's 100%. Right. And so it's possible that sort of the access to cooler waters could be mitigating in some ways. But it's not that it's basically if you have enough susceptible individuals and the disease present, you can end up with just so much disease that it doesn't really matter that it's not optimal temperature for the disease, as long as it's not like below their minimum threshold.
Okay, well, that you kind of answered a question that was coming up there. So we'll skip that one. Are sunflower stars territorial? And two, what is their range?
Yeah, I would say that they don't seem to be territorial. They certainly are all together a lot of the time. But I will also say that they are not taggable. So one of my colleagues described them as loosely held together, which is maybe my favorite way to describe a sunflower star. They're like, they really can just drop an arm. They drop arms because of this disease. They can also drop arms just because they drop arms. And so if you put a tag through maybe part of their arm, they will just drop it and walk away. And if you put a tag inside their body, it'll just get ejected. And so it's really hard to tell individuals, which makes it hard to learn anything about how much they move, where they are. Like, we know they move quite a bit, but we don't know how much. Are they just moving around in one place, or are they moving? There's like a lot of open questions because it's really hard to tell individuals apart. There's some pretty cool new work that's coming out that's trying to use AI to tell individuals by taking pictures of the top of them, because they do have some variability. So maybe we'll learn some things.
Yeah, especially as the population is sort of decreasing, and the ones that are the survivors, like that's kind of, to me, one of those things. Like there was with COVID, some people seem to have almost like a natural immunity. If they got sick, they didn't get very sick.
Yeah.
And it seemed to be an individual thing. So, genetically, maybe, there's some out there that don't get as sick.
Yeah. And that's again on the list of things we're trying to do, is see if we can find individuals that are more resistant in the sunflower populations. The other question was, what's their range? It's actually from Mexico to Alaska. So that map of the loss is the entire map of their range. So that, yeah.
Wouldn't find them in the Western Pacific, Japan, Russia.
No.
All right. Is this found in any other echinoderms, like urchins, and has anything else been susceptible to it?
Yeah, so urchins have their own array of mysterious diseases. So there are a bunch of unknown things that are happening to urchins, and we don't know why they're dying. But they don't seem to be happening at the same time or in the same way as the sea star die-offs, as it were. So like when we have a big sea star die-off in a location, the urchins didn't all die, right? And so it suggests that that's probably not an issue for the urchins, but we've got to look more. And we certainly, have a lot of work to do figuring out all the rest of the unknown diseases.
All right. So Kelly is asking, to the best of your knowledge, how far up the food chain has this disease affected other animals? Or has it?
Yeah, so to the best of our knowledge, nothing else is dying from VPEC, but that doesn't, that's to the best of our knowledge, right? So the effect to other animals in the food chain is mostly the loss of this big predator. Unless that, to be fair, not a whole lot of things eat sea stars. Gulls eat ochre stars, but there's not like a huge amount of predation on a sunflower star. So there's not much on the top side of that particular food web. The other thing is that we have some idea of the upper thermal tolerance of VPEC, and it doesn't tolerate being in a mammal. Like, mammals are too hot. So that's good news for us. And probably suggests that, I think most of the things that might be consuming sea stars would be in the mammal group, and so they probably aren't susceptible. But certainly one of the things that we are doing now is looking in as many other species and things as possible to see where else VPEC is. So like, where is it in the water? What's the variability? Maybe is it on or in mussels, for example, because sea stars eat mussels. And so that could be a good way to get the disease. So that's, yeah.
So because it was detected initially in France, you said in bivalves, I think scallops and another
scallop and oyster larvae.
And oyster larvae. So could this outbreak in British Columbia, say, affect the oyster farms and so on?
I mean, that's an excellent question. I don't know the answer to that question. We would need to do, so one of the things that's interesting about bacteria is that, so what we found so the VPEC we got from sea stars, we named it VPEC FHCF3, which is like a strain. So it's a specific, basically sea star VPEC. And then there's scallop larvae VPEC. And while they are both Vibrio pectenicida, like if you look at the genetics, they are Vibrio pectenicida, whether or not they cross contaminate each other, like whether or not a scallop one would make a sea star sick or a sea star one would make a scallop sick, like we don't know. We would need to actually do experiments to test that. Just because Vibrios change so much and so easily while staying the same species. So they're a tricky little bug. But one thing, one of our little tales of information that we're looking at is the one place that VPEC has been detected was from a gooey duck larvae, either in Oregon or Washington. And the one from a gooey duck larvae did look a little more similar to the VPEC that we have here than the one in France. So like, that's interesting. And we will continue to try and figure out that. But it's, that's like I said, it's on the long list of things that are sort of super exciting and intriguing. And we don't know yet.
Okay. You mentioned reintroduction programs. How are they going and where?
Yeah, so the work that's being done on that is primarily in the United States. There's a lot of focus on it in California because they lost like 90% of their kelp forests, and they really have this feeling that having and 100% of their sunflower stars. And so they're really focused on trying to reintroduce there. So the Cal Academy of Sciences has a program. The San Diego Zoo Wildlife Association has a program. There's a group called the Sunflower Star Lab that have a program that's all in California. And I think there's more I'm probably missing. And then the work was actually originated and like when this happened, we did not know how to raise sunflower stars. And so a researcher named Jason Hoden at the Friday Harbor Labs developed how to raise sunflower stars, and now it's possible for all these groups to do. So there are some folks raising them at Friday Harbor Labs, and then I think they might be starting something in Oregon as well. They've done trial outplants, and yeah, to various successes. But we are working with the Nature Conservancy to see if there are ways that we could start to try and breed for resistance to the disease, because I'm certainly quite concerned about putting sunflower stars that are super susceptible to this disease back out and having them die. And so if we could help them get a little less susceptible to the pathogen, that might make those recovery programs more successful. So that's one of our big goals.
Yeah, Jason did do a talk for us probably about 4 years ago, maybe 5 years ago, on the work they were doing at Friday Harbor Labs. It was still in its infancy back then. Yeah. Still kind of working on it. Sheila, you had a question about the selenic fluid?
Yeah, just throwing out there that I saw a paper recently out from DFO, I think it was, on cultured oysters being affected by a Vibrio, I think I've got it right, Estuarinuranus. So it was a Vibrio species, and I think that was fairly recent. So there's something going on there too. And when I'm thinking of clusters of oysters and clusters of sea stars, like there's some population abundance thing happening there. However, coelomic fluid. So as you were talking, I'm going, could the effect of the Vibrio be enhanced by the hydraulic system? Because it's pumping through so much water, if there's already a lot of Vibrio existing in the surrounding waters, is it concentrating those Vibrio populations to the point that it's the pathogen or whatever it is, toxicity, is more effective that way. But then you explained about the dermal swab and the tube feet, and I mean they're outside all the time and they're moving around. So then I was thinking, well, maybe that doesn't really matter with how much they're pumping through their body. It's just there.
I mean, I do. So the fact that sea stars take seawater in from their surroundings into their body regularly feels like a pretty easy transmission mechanism for pathogens. I mean, they must actually be very good at fighting most pathogens, because
Right.
they're constantly taking in seawater straight into their blood, because that's what their blood is, right?
Yeah.
So, but honestly, that's how I assume VPEC is getting in there. I don't know.
It would be hard. It would be very hard to test that, I think.
Yeah.
And that's when, when I saw the information on the tube feet, I'm going, well, they're outside all the time. So, I mean, if they're only getting affected a small amount or a larger amount, depending on which surface you're testing, unless what they're taking through the hydraulic system is finding areas to how to explain it, to hold on, like to stay inside kind of thing, like and building the population of the Vibrio that way. But then how do you test that? I don't know. Anyway, that's just something that crossed my mind.
Well, that is all of the questions. Oh, one more. Is Vibrio an established species or newly found? Vibrio.
This Vibrio. This Vibrio is an established species. So it was not a new one.
Okay. We're just coming up to 9 o'clock. There's no more questions in the chat, but just sort of as how you put, kind of looking into the future.
Mm-hmm.
Do you see anything that could help in the future to give sea stars sort of that chance, the sunflower sea stars in particular, the chance to come back?
Yeah, I mean, I hope that all these people who would like to try and recover them, like we do have a decent group of people who are interested in doing the work. So I'm hopeful that will be useful. I think the thing that I see, the most hopeful thing that is happening is that in the last year or two, people have started to see sunflower stars in the wild in California.
Oh.
So, we're still down to like, I think 20 have been seen, which is, not the billion that used to be there, but it's certainly more than the zero that was there for the first 8 years after we observed them, right? And so the fact that it's a species, they broadcast spawn, so they release their larvae into the water column, and those larvae can feed, and they actually can clone. They're really cool. Like this sort of larval stages are really interesting. And so I think the thing that's most hopeful is this idea that maybe, and maybe there's some place that all the larvae are just waiting and they're going to come recruit back in, and once the conditions are good. I don't know that's, I don't know. I think the way I like to think about it is I don't like to bet against somebody with that good recruitment system.
Mm-hmm.
And so that's the thing that gives me the most hope. And if we are able to find some resistant ones, then I'll get even more hopeful.
Yeah, sorry, I was just going to say, is it feasible that this whole event that's been going on, that with the Vibrio, that the larvae are actually adapting to this new environment? I mean, that's getting genetic, but it could be a slow adaptation through time as well, that they're able to adapt or to resist the effect of the Vibrio.
I mean, I would love that to be true. I don't know.
On the plus side, I saw a big one from the ferry on Quadra.
Nice.
Looking over the side going, whoa, look at that sunflower star.
Oh.
It It is 9 o'clock, and so we promised you we wouldn't keep you past 9 o'clock.
Yeah.
Like comments about, fascinating talk and so on. Thank you for sharing that with us. Sometimes we hear the results of, oh, this is the answer. But you never really see that journey of how it got there. So thank you so much for sharing the journey with us, and that story and your research and all the amazing information and so on. So
Thank you so much for your interest and for having me.
yeah.
Very nice.
Awesome. Here come the hands, class.